So, title is Nucleic Acid Structure, specifically focusing on RNA. In our previous video, when we talked about nucleic acid structure, we talked about the elements of nucleic acid, but we really focused on DNA. And we touched briefly on RNA, but now we'll focus on RNA specifically. This is the same image that we used when we were talking about DNA. So this is a nucleotide. And nucleotides are also the building blocks of RNA. RNA nucleotides have a few key differences, which is what we're going to talk about. The first, which is surrounded in the red box on your screen, is which of these nitrogenous bases can be incorporated into RNA nucleotides. In RNA, uracil takes the place of thymine. So if you look in that red box, over on the left is RNA, over on the right is DNA. And you can see that the base, so the nitrogenous base, is labeled uracil in RNA. So right here, uracil. And in DNA, that's thymine. So in RNA, rather than CGAT, you have CGAU. So if we had a strand of DNA that was adenine, guanine, cytosine, thymine, thymine, adenine, adenine, guanine, what would be the complementary base pair in RNA? So what would RNA be the complement to this DNA? So something that sometimes helps, depending on the way your brain works, is sometimes it helps to first figure out what would the complementary DNA strand be. So adenine always base pairs with thymine, guanine always base pairs with cytosine, cytosine always base pairs with guanine, thymine with adenine, adenine with thymine, adenine with thymine, and guanine with cytosine. So now we've figured out what would the two strands of DNA look like. So this is our strand that we're trying to figure out what would the RNA base pairs look like. So we call that the complementary strand. What would the complementary RNA strand look like? And so sometimes an easy first step is to figure out what the complementary DNA strand would look like. So that's what we did here. This is DNA. And then from there, it's really easy to figure out the RNA. So using purple to represent RNA, as I did with the arrow. So what's different between RNA and DNA? What's different is thymine gets replaced with uracil. So now what we need to do is just look for our T's. So our T's, instead of thymines, they're going to be U's. They're going to be uracils. That's the only thing that's going to be different in the complementary strands of DNA versus the complementary strand of RNA. So that's how we can figure out what our complementary RNA strand is by first figuring out the complementary DNA strand. Of course, you don't have to do that. Totally cool if you don't want to do that, but it's a helpful step, okay? And also understanding this complementary DNA strand is going to be useful in a lot of applications as we move forward in analyzing genomic information. So now let's take a pause. So I showed you kind of how to do that. I'm going to give you another strand. So what if our strand is what if this is our DNA strand and we want to figure out our question is, what's the complementary RNA? Okay, so I'm going to give you a second to figure that out. And then a question's going to pop up here in a second. So if you need a little bit longer, go ahead and pause the video. Okay, so you can do this different ways, but if we do it the way that I showed before, let's first figure out what our complementary DNA strand is. So what DNA nucleotides would base pair to this strand? So we would have an adenine pairs with each of the thymines, a cytosine with the guanine, a guanine with the cytosine. Sorry, I'm writing sort of upside down. Thymine, guanine, thymine. Okay, and so then for figuring out our RNA, all we do is we replace our thymines with uracils, and everything else is going to be the same. Okay. Okay, the next major difference between DNA and RNA is the 5-carbon sugar. So recall that the orientation of this sugar determines the direction in which a nucleic acid strand runs. So whether it's running 5' to 3' or 3' to 5'. Sorry, I hadn't switched the screen. So we're looking at our sugar, our 5-carbon sugar, or our pentose sugar. So pentose means 5, pent means 5, like the pentagon. So this 5-carbon sugar is different in RNA than it is in DNA. So here I have a visual of the 5-carbon sugar in DNA on the left and a visual of the 5-carbon sugar of RNA on the right. So just take a second and see if you can identify any differences. Okay, so if you look at the 2' carbon, whoops. So remember that the numbers, the primes of those carbons are really, really important. And if we look here at the 2' position on each of these pentose sugars, what we see is that in DNA, there's no oxygen. Whereas in RNA, there's an O there, that's an oxygen. Okay, so just like the one next to it. So here and here, both of these have an oxygen. Whereas in DNA, the 3' has an oxygen, but the 2' is missing an oxygen. So in DNA, that oxygen is missing compared to the sugar in RNA. The O denotes oxygen. So this is where this sugar gets its name. So in DNA, this is deoxyribose sugar. That's the name of this sugar. Deoxyribose. And so look here, deoxy. So that sugar has lost an oxygen. It's been deoxygenated. Okay, that's where its name comes from. Whereas RNA just has the ribose sugar. It hasn't been deoxygenated, it's just ribose. And so this is where their names come from. Deoxyribonucleic acid or ribonucleic acid. So this is a really, really critical and important difference between DNA and RNA. Also, I realize I say critical too much and I apologize for that. It's just, I'm working on it. Okay, so the final major difference is that RNA is single-stranded, whereas DNA is double-stranded. So you're familiar with the DNA double helix. So that classic double helix structure, we see it a lot. Okay, its DNA is a double helix. It is double-stranded. Oh, I can't spell. Helix. Okay? It's double-stranded. Whereas RNA is not a double helix, it's single-stranded. Okay? So RNA is single-stranded with exceptions. Okay? One of the most exciting, fun, awesome things about science, as we discussed at the very beginning, is that it's a process. Okay? It's a process of learning and understanding. It's also not as cut and dry as we want it to be. Okay, so we as human beings have a tendency to need to categorize things to understand. But keep in mind that these human categorizations of the natural world couldn't care less about our human need to make things fit into conceptual boxes. So when we're talking about the natural world, there are almost always exceptions. Okay, so RNA is single-stranded. This is one of the primary differences between RNA and DNA, but there are exceptions. So I'm really going to encourage you throughout this course to get comfortable with the gray areas. Get comfortable with the fact that there are so many gray areas in the natural world. And this is where the magic often happens, is in these gray areas. Okay, so RNA is single-stranded, but many types of RNA fold onto themselves to form their functional structures, much like a protein does this. And we're going to see some images of that in just a moment. So I'm going to show you this video, which is really cool. It's actually just came out, and I've given you the link to it. So this is a three-dimensional model from a recent research paper where researchers are seeking to understand how exactly it is that RNA folds itself. And the headline refers to it as ties itself in knots. And so what researchers are trying to understand is when RNA folds itself, is it able to unfold itself and fold itself on its own, or does it need helpers to do that? Does it need helper proteins? That's the subject of that research paper. And so this is an image kind of showing you how RNA can, or a video I should say, of how RNA can fold itself. So RNA is single-stranded, but many types of RNA do need to fold themselves. Okay, some viruses have double-stranded DNA. Viruses, you guys, are like the quintessential blow your mind, gray area, doesn't fit in any boxes, not interested in human categorization. So viruses, are they living? Are they non-living? We don't know. Okay, they simply don't follow the rules of other life on Earth. So one of the rules they don't follow is the single-stranded RNA thing. Some viruses just naturally have double-stranded RNA. And one of those viruses is the rotavirus. And the rotavirus causes childhood diarrhea. Almost every human child on the planet gets rotavirus, and almost every human child gets rotavirus multiple times during their childhood. And the immunity to this virus increases with each subsequent infection. So to the point where when you're an adult, you almost never get an infection from rotavirus. But this particular type of virus is double-stranded, has double-stranded RNA. Okay, of note, because of the times and when we are, where we are, SARS-CoV-2 is a single-stranded RNA virus. Just FYI, specifically, it's a plus sense single-stranded RNA virus. And I've given you a link here if you'd like to learn more about positive strand RNA viruses, because this is so valid right now to want to understand more about this virus that's causing this massive pandemic. You'll notice that I've given you a Wikipedia link. So I don't know about you, but it seems like students are told, do not go to Wikipedia. Wikipedia is not a valid source. So let's talk about that. I'm going to take you to this page. Okay, so here is the Wikipedia page that I gave you the link to. And just want to show you, I don't know about you guys, but I'm guessing that you hop over to Wikipedia fairly often when you need to understand something. And I want to show you something. So the reason I think that often we get told don't go to Wikipedia, or you get told don't go to Wikipedia, is because it is not a primary resource. And we already talked about primary resources and why they're reliable, because of the editing process and then the scientific community testing over and over again, things that get published in the primary literature. But if we scroll down, here's that page. Down here at the bottom in the references section, this page is so heavily referenced. And if you look, you can see that these references are to the primary literature. Okay, you've got bacteriological reviews, the Lancet, Nature reviews. Okay, that does not mean that you automatically believe everything it says, but what that means is that this page can serve as a jumping off point for you to learn more from the primary literature. Okay, it can also serve as sort of an entry point to understanding what the heck the primary literature is talking about. Okay, so that's how Wikipedia pages can be really, really, really useful. Okay, so here, this is a summary of what we've talked about so far with how RNA is different than DNA. And we're going to finish up by talking about the classes of RNA. And this first group are the classes that you are probably really, really familiar with. So the first one is messenger RNA, mRNA. And this image, what this image is showing you is kind of the two types of mRNA. And they're, oops, didn't mean to do that. Let's try that again. And we're going to talk more about these. Why are you not, dude? Okay, we're going to talk more about, a lot more actually, in multiple layers of lectures about what pre-mRNA is and what mRNA is. But you're familiar with mRNA because you learned about the central dogma, transcription and translation. And mRNA is how DNA is transcribed. Okay, DNA is transcribed into mRNA. Next, tRNA, this is transfer RNA. So tRNA is critical to translation, and we will talk about tRNA more when we talk about translation. Point out that tRNA here, it's showing, this image is showing that folding, how we have a single strand of RNA, and then it's folded back on itself to where there are places where it's base pairing with itself. Okay, so there are places where that tRNA is double-stranded. Then rRNA, rRNA is ribosomal RNA, and this you just have sort of two images here to show you two different versions. Once again, like is going to be a consistent theme, depending on what characteristics of these incredibly complex structures we're talking about, you're going to see them visualized different ways. They're both accurate. In the image to the left of the rRNA that's spinning, that's the rRNA complexed with proteins to form the ribosomal subunit, which we'll talk about when we talk about translation. So the purple parts are proteins and the sort of yellowy parts are RNA. And then over on the right, this is showing one particular type of rRNA in its two-dimensional structure, so how it's folded up on itself, highlighting again that very frequently RNA is going to fold onto itself and base pair. So those are the ones that you're probably familiar with. Types that you're probably not familiar with, maybe have never even heard of before. So snRNA, and on the next slide, I list the full names of all of these so that you'll have those. So snRNA, what does it do? It processes that pre-mRNA into mature RNA. And we'll see a lot more about that when we talk about transcriptional regulation. Lots of different miRNA, siRNA, piRNA, LNC RNA, names again on the next page. These regulate gene expression. So you can imagine if these are regulating which genes get expressed when, they have incredible control over our bodily functions. And some really detrimental diseases have been linked specifically to malfunctions in these RNAs. Then telomerase RNA. At the end of chromosomes are these sections called telomeres. They have to be replicated in a very specific way. There's an enzyme called telomerase that does that, and this RNA is necessary for telomerase to do its job. So the take home, as of right now, until we get into the details of these, is there are a whole lot of different types of RNA, and they do a lot of really, really important jobs, everywhere from actually transcribing DNA to regulating transcription of DNA, from translating mRNA to regulating the translation of mRNA, and also they're necessary and critical to DNA replication. So here are those names as I promised, so that you have them on the last slide, and you have the PDF of this video. If you look just above where you're watching it, the PDF is there, so you have those written down.